So it really gives me a great pleasure to welcome you to Stanford Pulmonary Hypertension Grand Rounds, or maybe better known as Stanford Penn Pulmonary Hypertension Grand Rounds. I think we have both centers involved. For those who don't know me, I'm Stephen Kawit. I've been a visiting professor here at Stanford now for 5 months on sabbatical, and I work at Penn, and it's really been a fantastic time. And it really gives me great pleasure to introduce Ms. Jackie Scott. So she got her bachelor's degree. I was just calling to see if I could reschedule Sarah's walk. Can everybody put themselves on mute, please?
Later time in the afternoon.
Can we mute all?
Yeah. 453575664.
Hang on one second. So let me go ahead.
Yeah.
Oh boy.
I think I saw.
There we go. All right, so it gives me great to introduce Ms. Jackie Scott. She got her bachelor's in bioengineering and her master's of science from Michigan in physiology. She's currently a PhD student at Carnegie Mellon, biomedical engineering. She was supposed to present her results at ATS. She actually won an abstract award. Unfortunately, of course, ATS was canceled, so we thought this would be a great forum for her to present her work. She's currently working at the FDA, looking at clinical trials, but has a really interesting background and has worked in a lab focused on ECMO, as well as worked at Fitbit for a while. So really a very interesting combination of skills and experience. So today she's going to talk about machine learning and pulmonary hypertension. In terms of the ground rules, I think it's best, first of all, for everyone to make sure that their microphone is muted. So please do that. During the talk, I'll ask you if you have questions to type them into the chat. And what we'll do, Jackie, is I will try to chime in and interrupt you gently for questions. Okay. And what we can do is at the end of the talk, we'll use the raise hand function, and then we can kind of open it up to call on people to ask questions themselves. So without further ado, Jackie, why don't you take it away?
All right, thank you very much for that lovely introduction. I think Dr. Kawat nailed it on the head. I have a very diverse and interesting background. So hopefully I get to share some of that with you today. And this talk will be touching on a little bit more than just my abstract from the ATS. So just giving you a heads up. So good morning or good afternoon or good evening for wherever you are in the world. As was explained, my name is Jacqueline Scott. I'm a PhD candidate at Carnegie Mellon University. And today I'm going to be talking to you about my research in collaboration with Ohio State University on enhancing clinical science with machine learning related to pulmonary hypertension and beyond. So just briefly, gonna touch on the learning objectives for this talk. We're going to discuss a bit the benefits of using machine learning over qualitative risk assessment for patients with pulmonary arterial hypertension. And then we're gonna discuss a little bit how we can start to bring machine learning into clinical science for these patients related to both clinical trial design and treatment guidance. And how we will be accomplishing that today is first, I'm going to discuss a little bit about the clinical challenges in treating patients with pulmonary hypertension, and then take a really deep dive into how machine learning can better support clinicians to improve treatment for patients, as well as improve clinical trial design. So without further ado, let's jump into the clinical challenges in treating PAH. So as most of you in the audience are already aware, pulmonary arterial hypertension is a rare but deadly disease. There are 5 total classifications for pulmonary hypertension, and I do really want to mention and focus on that my research is primarily WHO group 1, which are patients who have inherent disease of the pulmonary artery instead of an external factor such as left heart disease. And so this is a small percentage of total PH patients, about 16%, with a prevalence of 29.1 per million. So to give you a little bit about the history of treatment for PAH, we have a number of unique drugs, and yet survival remains poor for these patients. So it was 1991 when the NIH registry first established that the untreated median survival for patients with pulmonary arterial hypertension was 3 years. And then in 1995, we saw our first drug approved, which was IV epoprostenol. And it took about almost 10 more years to sort of realize the effects of that monotherapy, which was published to improve the median survival to 5 years, with oral Bosentan and subcutaneous treprostinil approved in between there. And then the early 2000s saw a huge flurry of different drug approvals. And in 2015, we were able to establish that the median survival improved to 7 years. So the moral of the story here is that we have a lot of treatment options, and yet we need to figure out if we can really manage treatment in an optimal way so that we can improve survival for these patients. And so to start to answer this question, the European Respiratory Society came out with a treatment algorithm that essentially said that accurate risk assessment is crucial for treating patients with pulmonary arterial hypertension. So this is a flowchart adapted from the European Respiratory Journal from those 2015 guidelines. Starting with a treatment-naive patient, you would assess their risk of death in 1 year, with low-risk patients being those with greater than 95% survival, so less than 5% chance of a mortality event, intermediate between 5 to 10, and high greater than 10. You would then establish what their upfront therapy would be with low or intermediate, possibly taking either an initial oral monotherapy or combination therapy, and high-risk patients starting right away on an initial oral combination plus IV prostacyclins. Or if your patient is already on treatment, you would assess whether they're having an adequate response to their treatment. And if that answer is no, you would escalate, either using sequential therapies or putting them on the waitlist for lung transplantation or considering an atrial septostomy. And so there are 3 major questions that come out when we look at this treatment algorithm. The first one is, what is the right tool for us to assess risk of death in 1 year for these patients? And I'll talk a little bit about the number of different risk calculators that are already present to try to help with this and their limitations. The next question is, can we actually be more precise if we're stratifying patients very accurately? Can we get rid of this ambiguity about whether we start low risk or intermediate risk on an initial combination or monotherapy? And the next is, what truly qualifies as an inadequate response? Is it that the patient was able to achieve a lower risk status, or was it that their risk went up? How do we quantify that? And can we use risk assessment to do so? So as I stated, there are a number of risk calculators that exist currently to try to stratify patients into a low, intermediate, or high. And they are informative, but they're lacking in precision and usability. So in this 2019 CHEST article, it was revealed that Reveal 2.0 has one of the most accurate and precise risk calculators currently used in clinical practice, with a C-statistic of 0.76, which is generally considered to be good. A C-statistic of 0.5 would indicate that the calculator is performing no better than chance. And most accurate calculators are sort of within that good or close to good range, with Compara and the French score of C-statistics with C-statistics of 0.62 and 0.64. But to really say that a C-statistic is great, we need to start achieving values of greater than 0.8. Further, these calculators don't intelligently handle missing variables. Reveal 2.0 is said that it can be used to risk stratify even if you only have 6 of the 14 variables, but it does become less precise when you have missing data. And further, these calculators don't reflect complex decision-making. Each variable is only linearly related to risk and can only be additive and subtractive, and there's no consideration about interactions between variables or the sort of higher level considerations that clinicians are trying to make when assessing their patients. And so how can machine learning start to answer this question of improving risk assessment? So first, for those of you in the audience that have some familiarity with machine learning, but don't quite know what is the difference between machine learning and statistics, I wanted to break it down a little bit. Machine learning is really a tool that's optimized for prediction. And so in statistics and machine learning, they're sort of two sides of the same coin. Both of them can be used for exploration of data. And I'll talk about this as I go through. For example, what are the clinical variables that predict survival in PAH? But statistics is really designed to confirm, test, and support hypotheses. And so an example of this would be in a clinical trial when you're trying to answer the question of what treatment significantly reduces PAH clinical worsening. Whereas machine learning is optimized to predict. So trying to answer the question of what is the probability of my patient dying or being hospitalized within the year. And certainly you can perform prediction with statistics. And there are some people who have started to look at ways of supporting hypotheses with machine learning, but they are optimized for different tasks. And so when we're trying to predict for patients, we want to choose the tool that's optimized for prediction. Specifically, my research focuses on Bayesian networks, which are a really powerful tool because they can map and visualize probabilistic relationships. So shown on this slide is an example of the most simple implementation of a Bayesian network, which is a naive Bayes, where we have our outcome, which is 1-year survival, and we have our clinical variables, mean pulmonary arterial pressure, cardiac output, and 6-minute walk distance, as the variables that we're trying to use to predict that survival. So in terms of sophistication, a naive Bayes is not that much more complex than the risk calculators that are currently used, as there's no interaction between variables. So we can start to look at one greater level of complexity, which is a tree-augmented naive Bayes. And now this is allowing for interaction between these two variables, which is mean pulmonary arterial pressure and cardiac output, which we know are affecting each other through the circulatory system. And so by allowing for this variable interaction, we can control for noise. So no one clinical variable is dominating in terms of predicting that survival outcome. And we can consider multiplicative effects, such as for a patient that has a high mean pulmonary arterial pressure, but a high cardiac output, that would be suggestive of a better outcome because their right ventricle is still functioning, versus a patient with a low mean pulmonary arterial pressure and a low cardiac output would be indicative of a worsening chance of survival. And so we considered a couple of different steps to go from traditional risk calculators, such as the Reveal 2.0 score, to FORA, which is Pulmonary Hypertension Outcomes Risk Assessment, which is a tree-augmented naive Bayes. And so starting with Reveal 2.0, we transformed this calculator into FORA 1.0, which is a tree-augmented naive Bayes network that has all of the same clinical variables as Reveal 2.0, and it also uses all of the same cut points or ways of breaking it down, continuous variables, to determine how they contribute to overall risk. And then we wanted to take it one step farther and take a really deep dive looking at all of the possible clinical variables that could predict survival and their interactions and learn an entirely new network. And this is what we call FORA 2.0, and this is the model that I've been working to develop for the past 2 years. So we're moving from this concept that's rooted more in statistics, specifically Cox proportional hazards, to a model that is more machine learning based from the variables we learn, the cut points we learn, and the final classification. So to provide some background on how FORA 1.0 was doing for us, simply converting Reveal 2.0 gave some gains over traditional risk calculators. And FORA 1.0 was established in the Reveal registry database, which is a US observational study. And it was able to achieve a C-statistic of 0.8 in its cross-validation versus Reveal 2.0's C-statistic of 0.76, pardon me, as shown in the receiver operating curve here. And these results were published in the European Respiratory Journal this year. Farther, and included in this publication is the validation of FORA 1.0 in the PH SANS registry, which is an observational study coming out of Australia and New Zealand, where 401.0 was able to achieve a C-statistic of 0.78 versus Reveal 2.0, 0.74. And then again, validating in the compare registry, 401.0 achieved a decent C-statistic of 0.74. And so we really wanted, and so seeing that this new model with variable interactions was able to provide us with a bump in the C-statistic, now we wanted to go through and really try to push this accuracy as far as we could. And that led us to build for a 2.0, which followed a process to avoid overfitting and really maximize the use of clinical variables with the most relevance and predictive power. So in that process, what we did was we worked with United Therapeutics and Bayer to do a meta-analysis in their clinical trials to find the variables that were the most significant in predicting survival. And concurrently, I worked at the FDA to harmonize 5 PAH clinical trials to provide a high fidelity data set for model training. And so variable selection from the meta-analysis came by the significance of the aggregated p-value using the Stouffer method. And then the harmonized data set was broken into a training data set and a testing data set. We then did farther variable refinement and selection, as well as discretization using the training data set and with the variables that we selected from our meta-analysis. For variables that we didn't have a large enough N for in the meta-analysis, but clinical experts still felt were very crucial, we included them in this process of candidacy for the model. Then went through and eliminated variables that were only independently predictive by chance, and I'll go into this a little bit more. Learned the network structure and its parameters, and then did our model validation on data that was completely unseen through the training process. So again, this testing data set is a random selection of this harmonized data set, and it wasn't part of any of the training.
Can I ask Jackie, so what was that unseen data set? Where were those data from?
Okay. So those 5 clinical trials included the patent trial, Seraphim, Griffin, Ambition, and I feel like I'm missing one, Freedom EV. Okay. So all contemporary trials.
And then the unseen data? That was another
The
trial.
unseen data is a random selection from those. So just randomly pulling out 20% of that data, using 80% of it for testing.
Gotcha.
Yes, thank you. So how important was it that we went through and did this meta-analysis? Why didn't we just do a literature review, ask some clinical experts what they thought, and then choose clinical variables based off of that? Well, we really wanted to try to capture, what is this relationship between how important a variable was from our meta-analysis versus how often is it mentioned in literature or by experts. And so we interviewed some experts that provided literature sources as well. And what we did was we mapped how variables had increasing statistical significance. And so this is the negative log of the p-value, where all of the variables to the right of this line have a p-value less than 0.05. So they're more significant. And all of the variables to the left here have a p-value of greater than 0.05. So they're less significant. As well as on the y-axis here, mapping the number of times an expert or literature mentioned that variable. And what we found was that specifically stroke volume index, stroke volume, and stroke work were the most significant hemodynamics coming out of our meta-analysis, and yet they were actually very rarely mentioned by clinical experts or literature for being predictive of survival for PAH. Whereas variables such as mean pulmonary arterial pressure were mentioned quite frequently, and yet were actually not significant from our meta-analysis. So this really highlighted the importance of using an evidence-based variable selection process for our model. And to give you sort of an even bigger sense of all of the variables that we considered for meta-analysis, this graph here shows a number of clinical variables on the x-axis Again, stroke volume index, and stroke work are included here, as well as a number of laboratory values and demographics. And then on the y-axis is their aggregated p-value. And again, variables that are below this line of p-value of 0.05 are more significant. Variables above this line are less significant. And so what was interesting about this is that there were a number of laboratory values that we found to be predictive of survival, such as sodium, blood urea nitrogen, and total bilirubin. And yet none of these variables are considered in current risk calculators. Or again, something like mean pulmonary arterial pressure, which is valued very highly, was not significant. So once we've gone through and chosen our candidate variables, we did this analysis to sort of farther reduce down the number of variables we're looking at, as well as to discretize our variables, because the modeling paradigm that we're using requires that you have categorical variables to do your risk prediction. So in order to discretize these variables, we used this process, which is an information gain analysis, which essentially tells you if I broke down my patients for all of them that had, let's say, a sitting heart rate of greater than 90 beats per minute, and those that had less, would I gain more information about what happened to them? So whether they died or lived at the end of that year. And so we went through, and with all of our continuous variables, either they were able to provide this significant cut point that improved the information or not. And then if they didn't, they weren't included. And first, we looked at a naive. So again, that simple no variable interaction model. But if they did provide a significant cut point, then we considered them to be farther candidates. And this cut down our total variables from 41, as I stated in the previous slide, down to 21. And so now we're able to visualize in this naive Bayesian network how the variables are related to the outcome. So the thickness of the arrows here shows you the strength of the relationship between that variable and the outcome, but independently of all other variables. So again, stroke volume here, very strongly related. Mean pulmonary arterial pressure, not very strongly related. Race, not very strongly related. And so the question was, now, can we reduce the total number of variables that we're looking at here farther, but can we do it in a way that's not arbitrary? Let's not just throw out the bottom 5 or the bottom 10, but let's try to think of a more objective process for doing so. And so that led us to this process of eliminating variables that didn't outperform their random surrogates. So what is a random surrogate variable? So I wanted you to consider this sort of toy example because it's kind of an abstract idea. So if you imagine having n total patients, and let's say you've discretized their NT-proBNP values into 3 categories. Either there's low NT-proBNP, average amount, or high amount. And so this is indicative of survival in this toy example where all of the patients with a high NT-proBNP did not survive 1 year. But now, if you considered a surrogate with the same number of cut points, but a completely randomly assigned value, you still might by chance get some amount of information from this surrogate. And so in this example, the 2 patients who had a low NT-proBNP value, but it's actually just randomly assigned, 50% of them died. 50% of them lived. All right, that's more information than anything else is giving me. I guess I'll go with that. So, and the other thing that's a slight step farther in the complexity of this is that as you have more cut points, so let's say now you broke it into deciles, and if you increase the number of cut points, the more informative a random variable would appear. So in this example now, even though the 10th decile is what's telling you whether the patient survived or not, in the randomly assigned value, just because this one is unique, it appears that the 4th decile tells you whether they survived or not. And so that's illustrated here on this graph. So we wanted to make sure that all of the variables that we were using were giving us a significant amount of more information, which we measured with this concept called J-divergence. And we wanted to make sure that it was better than just a randomly assigned value. And so we found that most of them are doing quite well, but race, as you saw in that image of the naive base structure before, is actually doing quite badly. And so we were able to eliminate this variable from our model based off of this analysis. So finally, we learn a final Bayesian network by learning the network structure first, which considers first the variable interactions, and then learns the joint probability for each variable pair and the clinical outcome, which is survival in 1 year. And so learning this network first required that the patients had all variables measured. So this was just a chunk of the total training set. But then we were able to refine parameters farther with this process called expectation maximization, which iteratively improves the parameters and maximizes likelihood of the model. And this doesn't require the patients to have all variables measured. So now we can learn from patients even if they have missing data. And once this final network is built, we can kind of look at it and tease out these really interesting interactions between variables. Excuse me. So the example here being 6-minute walk distance, which is related to etiology. And patients that had a low 6-minute walk distance, but had an idiopathic PAH, had a 1.2 times chance of dying over living, yet patients with familial PAH and that same low 6-minute walk distance had a 4 times chance of dying. And so this is just an example of those sort of multiplicative effects that you can glean from a model like this that you can't do right now with traditional risk calculators. And so what did this buy us? So the final model outperformed several other risk calculators on the same holdout test set. And again, this is a test set that is a random selection of subjects from those 5 contemporary trials. And for a 2.0, which is the model that I've just showed in the slide before, achieved a C-statistic of 0.83 versus for a 1.0, achieving a C-statistic of 0.77, revealed 2.0, a C-statistic of 0.76, and Compara and French, not achieving C-statistics above 0.6. So to give you a quick summary of all of the items that I've touched on, the process for building our model was to adapt Reveal 2.0 into a Bayesian network, which was 401.0, and this was built using Reveal registry data. We then validated 401.0 in external registry data, which is the PH-SANS and Compara dataset, which gave us good but not perfect performance with a C-statistic varying between 0.74 and 0.8. We then built for a 2.0 using contemporary clinical trial data, and then validated for a 2.0 versus multiple other risk calculators in these unseen randomly selected contemporary clinical trial data, and it outperformed all other models with an AUC, or rather C-statistic of 0.83. AUC and C-statistic are interchangeable terms.
So Jackie, I have 2 questions before you move on.
Yes, go ahead.
So one from Roham is the training analysis specific or corrected for therapies? Yeah, so how are therapies incorporated in?
I love that you asked that question, because that was the next point that I wanted to get to about future model development.
Okay, so we'll hold that one. And then the other question is there work trying to move beyond discretizing the variables? That you're obviously losing a lot of information when you're taking a continuous variable and cutting it into yes or no. Is that something in the field that's ongoing?
Right.
Or?
Yes, that's absolutely true. So actually for some variables, they're not so perfectly calibrated against the outcome that you lose that much information when you discretize. Truly, sometimes high and low is good enough to tell you something. But there are other types of Bayesian networks, specifically these mixture models, that can consider a variable that is modeled as a Gaussian distribution, so you're able to keep it continuous, as well as using discrete variables in the same model. The current software that we're using, it's mostly just a software limitation, requires us to discretize, but that is something that we are considering in the future, and was actually discussed in our grant and renewal for a 2.0. Yeah. Right. All right. So to answer that question, how do we consider background therapy? Well, we were going through and doing our steps to validate the model on other clinical trial data, as well as existing registry data. So highlighted here is sort of where I am in the process in these most recent months. And the really interesting finding that came out of this was that FORA 2.0 is doing better in data that comes from after 2015 clinical trials. And FORA 1.0 was doing better in clinical trials that were pre-2015. And specifically, the two other trials that we looked at were ARIES, which is the first approval for ambrisentan, and FIRST, which I believe is the first approval for tadalafil. And FORA 2.0 wasn't doing too badly. It was sort of a C statistic just shy of 0.8, around 0.76. But then FORA 1.0 was doing really well in these trials, like AUCs of 0.85 and 0.8. And so that made us start to consider, well, there is a difference in background therapy from year to year, era to era. Do we need to start capturing that in our model? So we're going to go through and expand our training data set, including those previous clinical trials, and then learn what the new crucial variables are for capturing therapy. And so some of the work that I've started to do regarding this, the first thing that I looked at was diuretic use. So whether a patient was requiring diuretics, does that essentially improve or suggest that they're in bad shape? And so I've gone through and done a lot of analysis to do sort of this fuzzy matching process to pull out all of the medications from these patients. And then once we go through that, we can start to really hone in on what are the medications or management styles that are really crucial for predicting survival. Great question, thank you. All right, so that encapsulates all of the things that I want to talk about the current process of building our new machine learning model. And now I'm going to switch gears a little bit into talking about how we can start to think about integrating these models into PAH clinical science. So improving risk assessment would give us a lot of different opportunities to improve clinical science and PAH as a whole. The first one being this concept of enriching our trials. So can we pre-select patients when we screen them that are more likely to experience the clinical trial endpoint, which is typically clinical worsening. And then again, we want to improve upon the treatment guidance provided by the European Respiratory Society treatment algorithm, which is we want to use a standardized way of assessing whether patients are deemed low, intermediate, or high risk. And then we also wanted to dive into this question of whether upfront oral combination therapy is truly required for patients who are low risk, and what are the trade-offs when we're giving them that upfront oral combination therapy. And then I'm going to give you a bit of a sneak peek of the automated web tools that my team is designing to guide clinicians on regular risk assessment and treatment escalation. So let's discuss a little bit the current problems with trials in PAH. So trials in PAH are becoming increasingly event-driven, where the primary endpoint is the time to clinical worsening for patients. And so now trial size and length, whereas previously we just looked at things like change in 6-minute walk distance over 12 or 16 weeks of being on our drug, the trial size and length are now determined by the event rate in the placebo arm. And so in this article in the European Respiratory Journal from 2019, I thought this image really strongly illustrated this point. Where you can see with these 3 more contemporary trials, Seraphin, Griffin, and Ambition, they had a much greater duration and had to use a higher sample size because they have too many low-risk patients who have too few events, and they have to wait a longer period of time, and they have to enroll more patients to try to get to that total event number. So we tried to answer the question, looking at this as a retrospective study, could these trials have been enriched with traditional risk calculators? And so one of the first things that we did was to go through and we pooled all of the placebo patients across these 3 trials, and we looked at whether current risk calculators could break down and actually now predict clinical worsening instead of predicting 1-year mortality, because that is now the end point of interest. And so what we found was, and this example is showing sort of a survival tree where REVEAL is the only predictor of clinical worsening. And then it's able to identify these 4 groups across the placebo arm that all have statistically significantly different time to clinical worsenings. And What's also very crucial for this concept of enriching clinical trials is that we need to be able to show that if a treatment is studied and found to be efficacious in a high-risk group, that that treatment could be bridged to lower risk groups. So just looking at the hazard ratios, we found that they overlapped for the pooled placebo and the pooled treatment, suggests that the proportional response in lower risk patients or very low risk patients is still good. They're still seeing a treatment benefit, but the absolute effect is higher in higher risk patients. And so this table shows you a sort of more exhaustive list of all of the algorithms that we considered, what their risk groups were when we did the survival tree analysis, the incidence rate in the placebo arm, the incidence rate in the pooled treatment arm, and then again, that hazard ratio to see if treatments were similar for low, intermediate, or high risk patients. And so when we did this analysis, we didn't control for how many risk groups the algorithm could find. We just did the analysis and said, allow it to find as many significant cut points as it can. And so Reveal 2.0 was sort of the most precise. It was able to identify 4 different risk groups, one of them being this very low risk group, which had a lower incidence rate than the low risk of French or the Compara or just New York Heart Association class. And then it was really interesting. We applied 401.0, so that first implementation of a tree augmented naive Bayes, to the same data set. And it was able to identify this very high risk group, which had this exceedingly high incidence rate of clinical worsening. And also very interestingly, had proportionally a different response to treatment. They actually saw a greater treatment benefit proportionally and absolutely versus the low, intermediate, and high. And so we don't quite know yet what this means. It could be a chance finding. This included a group of about 40 patients across all of those pooled clinical trials. But it does suggest that maybe we could start to get more precise with machine learning. Maybe we can identify these very high-risk patients that might be seeing treatment benefits for certain drugs where lower-risk patients wouldn't see a treatment benefit at all. And then finally, to sort of wrap up this concept of enrichment, we had to answer the question of, is this actually cheaper to do? And the consideration here is that if you are looking for a high-risk group in sort of an already rare disease, now you have to screen for a lot more patients and you're only enrolling so many of them. So what is sort of that ratio of how much you have to spend in screening versus how much you get to spend on the clinical trial itself? And then does that translate to an actual potential net savings? Because you still get to enroll fewer patients, you get to treat them for a shorter period of time, and the drugs are very expensive in PAH. And so does that mean that you get to save money in the long run? And so what we found was that if you take this strategy of enrolling patients that are identified by Reveal to be intermediate and high risk, you're able to screen very, very efficiently because this group of patients isn't that rare versus just doing high risk only. And that allows you for a 40% net savings. And this is relative to a non-enriched trial. So if they had to use the same sample size, specifically, this is looking at only Griffin, the Griffin clinical trial. If they had to use the same sample size and the same treatment time, this is the savings that you would have versus that alternative. So the summary here is that you can enrich trials, you can use fewer patients, they can spend less time in the trial, less time on the placebo arm, which means we can get them to an effective treatment more quickly, and your trials can be cheaper. So this is feasible on multiple fronts. And then next, switching from clinical trial enrichment, now back to PAH therapies, we wanted to answer the question of what is the trade-off in treating low-risk patients with combination therapy? And so we did this retrospective study in the Ambition clinical trial dataset, which is from 2015. It had 500 treatment-naive patients. 50% of them were on combination therapy, 50% on pooled monotherapy. And we applied 401.0, so again, that first implementation of the tree augmented naive Bayes to risk stratify our patients. And we defined the low risk category as having a more than 95% chance of predicted 1-year survival, so less than 5% chance of mortality, and high risk as a less than 90% chance of survival, which would mean a greater than 10% chance of mortality. And because we want to understand the risk-benefit trade-off, now we're looking at a slightly different endpoint where we're considering the time to the first clinical failure event or the time to the first adverse drug event, specifically that causes the investigator to withdraw the experimental drug from the patient. Because these events are considered sort of the most severe. There's no reconciliation in terms of you can't put the patient back on this therapy if they're having some sort of hypersensitivity event or if they're having some sort of liver event. So we wanted to answer the question of, is this harming patients more than it's benefiting them? And so what we found was for the low risk patients, there's no significant benefit nor harm of being on combination therapy, which is the solid green line here. And you actually see quite a few early adverse events for those low risk patients. And then not much of a benefit in terms of reduction of clinical failure over the long term. Versus for the high risk patients, you still see that early adverse event rate, but then overall they start to be doing much better. And so this really begs the question of, is it that important to put low risk patients on combination therapy instead of considering sequential monotherapy? You start them on monotherapy, and then if they don't respond, you're able to elevate their treatment later. So in terms of how machine learning can start to be brought to the bedside, these are some screen caps of web tools that my team is designing to provide some fast, intuitive clinical use of machine learning models. So this is an example of what a clinician might see. They're able to put in the clinical variables for their patient, and then they would get a prediction of their 1-year survival, and they could compare it across multiple risk scores. This also has the opportunity to better guide treatment and better inform patients. So this is a possible dashboard on the patient side where they're able to visualize their current risk level, how their risk level is changing over time, as well as what were the treatments that were considered and on what day, and how does that sort of correspond with changing how they feel, how it changed how they felt. As well as considering, how rare am I? What are other patients looking like in terms of their risk? So in summary, we've taken this deep dive in machine learning, talked about ways that it can benefit clinical treatment of PAH, as well as benefit informing and educating patients about their risk status, as well as improving the dialogue between clinicians and patients. And lastly, we've talked a little bit about how it can be used to improve clinical trial design. So I just wanted to go back to that flow chart earlier from the European Respiratory Society guidelines and talk about how it can be changed with machine learning. So again, starting with that treatment naive patient, you're going to assess their risk of death in 1 year, but now you're going to use a Bayesian network with a good or a great t-statistic to do so. You're going to risk stratify those patients, and now we can be on a more clear initial track for them because we are assessing low-risk patients very accurately. We can start them on an initial oral monotherapy and then see how they respond. And now our inadequate clinical response is replaced with this concept of maintaining patients at a low-risk status or trying to reduce them closer to a low-risk status. And so if we don't, and so now we have a risk-based way of determining whether a treatment is efficacious. And if it isn't, we can consider treatment escalation, and that might look like funneling them back into this process. It might look like taking a slightly different step, but this is sort of an imagination of how we could start to really improve this treatment algorithm. Okay, so I just wanted to acknowledge all of my mentors on this project, as well as the Fora team, which is pictures on the top here. This project was sponsored in part by the Oak Ridge Institute for Science and Education, who funded my fellowship at the FDA, where I am mentored by Christine Garnett and Norman Stockbridge. Dr. Raymond Benza is the PI on the Fora NIH grant. Bayer, Actelion, and United Therapeutics all also actively contributed to our analysis, and I would like to thank my research advisor at Carnegie Mellon University, Dr. Keith Cook.
Great.
Okay, thank you. I'll now be taking questions.
That's great. I think Roham, you had a question. Do you want to unmute yourself? Oh, well, let me read it. So maybe he's got disconnected. So he had written a question, was concerned about enrichment risking generalizability. So in your mind, how do you balance those two things? And at what stage in development would you want to use enrichment?
Right. I think that is an important question. And it was something that we were also considering when we got the feedback on the manuscript. One of the things that was brought up was, well, what if you had a drug that actually you really needed for the early stages of PH? You would actually use an opposite approach. You wouldn't be enrolling high-risk patients, you'd be enrolling low-risk patients. And I think that's an absolutely valid point. But I think that it sort of bears to mind that you still need risk stratification then to really determine whether the drug is efficacious with a smaller sample size. And generalizability overall is a concern. In working with Norman Stockbridge and Christine Garnett, we are trying to kind of answer that question of, well, all of the drugs that we looked at for trial enrichment, we were really just considering essentially vasodilators, which we would think physiologically would have a similar effect across whether you're low risk or high risk. And so when you're trying to answer that question, you have to consider the physiology or the pathophysiology of the drug and what it's really doing for that patient. In terms of where that falls well within the process for clinical trials, it might be something that you start to consider if you have some pilot data from phase 2, but yeah, it really, it's going to vary by trial to trial. Great question.
Great, thank you. Others, and if you're shy, you can type in your question.
I could, thank you, it's Francois. This is an amazing talk, thank you. It was really good. In developing, one question I have, when we develop these models, you're developing it with mortality, but if you develop it for clinical deterioration, it could be different. So one first question, we often quote that a C statistic of 0.8 is acceptable, but for mortality, it could be actually good because we always have survivors. Despite worse clinical characteristics, if you look at 1 year or 2 years, they'll still be survivors. So first question is, what C statistics do you expect for mortality studies that are short-term? And the second question, developing models for clinical worsening versus mortality may be different parameters and different ways. And how would you approach that?
So let me answer your first question, and then I'm probably going to have you repeat that second question, because I was thinking about your answer for the first one. And I have a graphic that I think might be a little bit useful for this. So this isn't for mortality, this is for clinical worsening. And so one of the things that we were considering for the clinical trial data is, all right, we have patients that are experiencing clinical worsening events, but they're not all censored at the same time. And actually all of the trials have different median censor times. And so we have to consider, if we try to standardize that time, what is the effect on the C statistic? And here it says AUC, but again, those are interchangeable when you have a binary outcome. And so actually, you said that if you have survival, if you're looking at survival in the short term, a C statistic of, less than 0.8 might be good. But what we found is maybe somewhat of the opposite, is that a lot of these risk calculators are actually very optimistic. And that if you start to only look at patients about survival in the short term, your C statistic might appear to be sort of inflated. And so if we looked at patients where we exclude patients that are censored before 1 year, and then look at that C statistic versus, just including all of them, the AUCs start to go down a bit. And then if you're looking at it longer term, the C statistics go down even more, which would suggest that they tend to predict that clinical worsening is not happening as often for these patients, whereas it might truly be. So in terms of what is a good C statistic for the outcome of interest, I actually think that that is not something that you can put a specific number on. I think what you have to consider is something like the Udens index. So what is your maximum sensitivity and specificity, and weighting that appropriately for what is the bad outcome of not stratifying patients. So if I choose to say, okay, I think that this patient is low risk when really they're high risk, and now I'm undertreating them, you know, that has a very huge implication versus doing something like, okay, I think that they're low risk, but actually they were intermediate, and maybe I do something slightly different, but it's not a drastic undertreatment for them.
Because you would put more cost then on false negative than false positive.
Exactly.
In your thresholds that you want to do for Bayesian.
Right. Yeah. And that question is something that we really do want to start to answer. You know, we kind of have looked at and focused a lot on C statistics, you know, getting above that 0.8, but then you want to start considering, you know, what is that Udens index for the task that you have at hand, whether it's treatment or whether it is clinical trial design and enrichment, because, you know, there's actually it's less of a cost of, you know, maybe trying to accidentally avoiding a patient and not enrolling them in a clinical trial versus undertreating a patient where, you know, that is serious harm.
If you use cost analysis and Bayesian approach for your criteria, then it doesn't become the Uden index, right? It becomes a criteria based on cost and Bayesian's approach, right?
It is something more, it is something more complex than the Udens index.
Yes, exactly.
And I think that in terms of terminology, I've kind of heard it referred to as, you know, a couple different things. Yeah, if you have a Udens index, but now you're also incorporating a cost penalty into that. So I guess I kind of been using that term as like an umbrella, but yeah, you would be.
The criteria, different criteria.
Different criteria, right.
Okay. No, thank you.
Jackie, I have a question if no one else has one. So, you know, so how do we implement this? You know, this is, I mean, it's awesome work. It's really a beautiful presentation. You know, so how do you get people to use this and convince them that it makes a difference?
Right. Yeah, that's a great question. And that's something that we've tried to start to answer with the for renewal. This is really great, you know, sort of optional graphic. But if you consider sort of these, let me go back to my image of the web tools, just to provide a background. Something that we also want to do with these web tools is that once we start to have buy-in from some clinicians in terms of using this, we can actually start to take that data and learn from it. And so if we start with sort of this baseline model of risk, and we use the treatment guidance from the European Respiratory Society, we then can start to stack up and see, oh, did that treatment recommendation from the European Respiratory Society actually significantly reduce risk for their overall patients? And if we wanted to look at that clinical site as a whole, how well are they doing in terms of patient survival and reduction of clinical worsening versus others? And so that is sort of the idea for the buy-in, is that if we can start to show clinicians, hey, this is how you're stacking up versus other clinical sites, and this is how much better your patients are doing because you're using this risk assessment model versus other clinical sites that are maybe using it, but are sort of mostly kind of taking it as a rough suggestion. Or I actually was reading this review paper that only like 30% of high-risk patients are actually treated with upfront IV prostacyclin, which I found really shocking. You know, it seems like it would make sense to use this aggressive treatment, but you know, there still is sort of this question of, you know, how aggressive do you really need to be? And so if we can kind of show clinicians that there is an overall benefit by incorporating that and into the web tool and rerunning that data, that I think that, you know, it can really start to provide a benefit for treatment.
Anybody else with questions? Okay, if not, Jackie, I really want to thank you so much for sharing really kind of spectacular presentation with, as we can see, multiple papers coming out. We're really glad and that you were willing to do this for us. And like I said, I think very, very novel stuff. So again, I appreciate your time and your willingness to share this with us.